I am Dr. Nikita Sharma, Associate Professor, Chemistry, SPNKS Government PG College, Dausa. Today we will start Unit 5th, Green Chemistry of M.Sc Previous. Today's topic is Synthesis of Nanomaterials. Green synthesis of nanoparticles aims at minimizing generated waste and implementing sustainable process. In recent years, green processes using mild reaction condition and non-toxic precursors have been emphasized in the development of nanotechnology for promoting environmental sustainability. Greener synthesis of nanoparticles represents an advance over other methods because it is simple, cost-effective, and relatively reproducible and often result in more stable materials. Microorganisms can also be utilized to produce nanoparticles, but the rate of synthesis is slow and only a limited number of sizes and shapes are amenable to the method compared to routes involving plant-based materials. In the method of green synthesis, there is no requirement for high pressure, energy, temperature, or toxic chemicals. Hence, nowadays many researchers are diverting themselves from using synthetic methods. Plants produce more stable nanoparticles compared to other means, and it is very straightforward to scale up. The risk of contamination is also lower. Hence, an alternative option for synthesizing nanoparticles is by applying plants and their parts. In the recent past, due to these advantages, green synthesis is gaining extreme importance in all the fields focusing on a greener environment. Extensive research work is carried out using plants and their parts for nanoparticle synthesis due to the ease in scaling up for larger production. Apart from being cost effective and environmental friendly, nanoparticle synthesis using green tea leaf extract of Terminalia sibula fruit, oolong and black tea leaf extract, banana peel, along with Cococacea esculenta leaves extract, sorghum bran extract, eucalyptus leaf extracts, and Tridex procumbens have been carried out. The use of emerging microwave assisted chemistry technique is conjunction with benign reaction media is dramatically reducing chemical waste and reaction times in several organic synthesis and chemical transformations. Magnetic nanoparticles. Magnetic nanoparticles represent an interesting material both present in various living organisms and usable for a variety of bioapplications. Nanotechnology involves the study, control, and manipulation of materials at the nanoscale, typically having dimensions up to 100 nanometer. This is a truly multidisciplinary area of research and development. The large interest in nanostructure results from their numerous potential applications in various areas such as materials and biomedical sciences, electronics, optics, magnetism, energy storage, and electrochemistry. Magnetic nanoparticles having connection to biological system and bioapplication usually exist or can be prepared in the form of either single domain or supermagnetic magnetite, various type of ferrite, iron, nickel, etc. Synthetic magnetic nanomaterials are most often available in the form of magnetic fluids, means ferrofluids, which have already found many interesting applications For example, sealing, damping, heat transfer, levitation of magnetic and non-magnetic objects, production of computers, loudspeakers, measuring devices, etc. The first magnetic nanoparticle necessary to create a stable magnetic fluid were prepared in the early 1960s. Micro and nano-sized magnetic support is recently developed revolutionary technology for bioseparation, especially for ligand fishing, protein, enzyme, DNA, RNA, and cell isolation or purification. Magnetic nanoparticles are referred to by various synonyms such as magnetic beads or micro and nano-sized magnetic beads. They are also called ferrofluids or magnetic fluids, meaning colloidal suspension of magnetic particles in a liquid carrier. Generally, these particles are part of nanotechnology, which can be defined as engineering of functional system on a molecular scale. It has the potential to create many new materials and devices with a wide range of application across the biomedical, chemical, electronic, and mechanical fields. Their supramagnetic properties have opened promising new perspectives for their application in several areas. The pioneering medical application in the treatment of lymphatic nodes and metastasis based on injecting metallic particles preheated in a magnetic field was first published by Gilchard et al. In 1957. Since then, magnetic particles have been modified by coating with antibodies, enzyme, proteins, or specific ligands that enable them to bind to other biological active compounds or receptors on the cell surface. In the last decade, magnetic particles have been increasingly used as a promising technique for a wide spectrum of biomedical applications. The growing interest in the magnetic carrier is focused on biochemistry, molecular biology, and medical specialties. Because of the superparamagnetic properties and micro and nano dimensions, magnetic nanoparticles can be used to isolate any target and linked to various manual and automated applications. Innovative research has produced a new application for magnetic nanoparticles that carries an exciting biomedical and bioengineering potential. For instance, cell molecule and nucleic acid separation, immunoassays, pathogen detection, protein purification, gene mutation analysis, and magnetic force based tissue engineering. Also, the interest in the potential application of the magnetic technique in pharmacy is significantly growing. It is currently being recognized that this magnetic nanotechnology could play an important role in this area.